Yes. Scientific study of behavior mental processing. So we talked about how the history came about, started out as mental processing, then because we didn't have the tools, it was like we could only study observable behavior. Well, now
I'm gonna use this to pretend like I'm writing stuff so he doesn't like call me.
And really, the interesting thing is the bottom line behind all of it, the reason that you're able to do anything that you do in terms of mental processing or in terms of behavior is because of biology. Biology runs the entire gamut. The reason you can laugh, smile, raise your hand, ask questions, go for a run is because underlying everything, you've got a biological substrate, a system of communication that allows for everything that you need. Even though psychology is a science in its own, it's completely dependent on an underlying biological networking system. Of course, you know that we have a brain that controls everything that we do. Do you know how it works? Which, trying to figure that out. That's one area that psychology has focused on. Neuroscience, neuropsychology, physiological psychology is trying to understand this phenomenon. Again, it's necessary to get anything. So to start with, I'm going to see what knowledge you already have about this area. So we've got a true-false quiz. So as quickly as you can, I'm not selecting it, but as quickly as you can, run through and answer these questions for me. True or false?
Damn it. Oh, he's not collecting anything.
Yeah, that's all right.
Okay, one is false because you didn't test that with DNA.
Oh, he's...
Two is false.
No, false.
Two what? Three is false. I think. They're all false. No, 7 could be true because it could be illiterate.
Maybe 7, yeah, maybe. But it's like, in the first class, he was like according to Sigmund Freud. Yeah, that's how it is. So,
You have 30 more seconds. All right, number one. Small amount of brain tissue from a person cannot be distinguished from that of a monkey. What do you think? Is that true or false?
false.
That is true. Like if you take a slice of brain tissue and you look at what comprises brain tissue, there's no
Well, you didn't say visual.
difference between those. What is in your brain?
This is
Same kind of cells are in the same brain.
This is what I don't like. They're not specific. They're too vague.
Number two, the human brain produces its own natural opiates that elevate living beings' pain.
False.
True. You may know what they're called?
Serotonin.
I heard somebody say.
Serotonin.
What are our own endogenous opioids?
That's right, no, because the last time he did it,
Serotonin is not an opiate, but it is a
he
neurotransmitter.
did questions like these, and he was like,
Dopamine is also a
false.
neurotransmitter,
Except
but not
for
an
one, and it was
opiate.
the according to.
Oh, he did one.
There's none of them proven.
He did one of this stuff for the very first class.
Are your own natural opiates.
I'm really
The brain accounts for a larger percentage of body weight in humans than in other species.
He's completely contradicting himself.
Now, that's that is false. some rodent-like animals. Electronically stimulating a caterpillar at a certain point can cause the animal to cower in the presence of a small mouse. That is true. We'll talk about an emotional area where that is in the amygdala. Both animals and humans seem to have pleasure centers located in the brain. That is also true. So you can look at animal behavior, like researchers have studied rats, and they found an area that if you electrically stimulate this area, which electrical stimulation produces the neurons to fire and stuff, they will choose pressing a bar to get electrical stimulation of that area of the brain over eating and other things. So, yeah. We ordinarily use only 10% of our brain.
False.
False. We'll talk about that in a little bit. Some people can read, can write, but are unable to read.
True.
It is true. And again, we'll talk about some of that later.
I mean, that
Some
is a
people have
given.
had hemispheres of their brain split with no apparent ill effect. Now, if you're like, I don't even know what that means, so just quickly, you have two hemispheres, the left hemisphere and the right hemisphere. Those two hemispheres are connected by a band of tissue called the corpus callosum. What this is saying is if I sever that connection between the two, would I see obvious negative effects? And the answer is true. Now, it's not that there aren't effects. They're not obvious. You can see them under testing, and we'll talk about some of those. Some of those are described in the lab. Neural impulses travel through the body at the same speed that electricity passes through a wire. That is, in fact, false. Early on, it was discovered that they used to think that that's exactly what it was. The way that your brain worked is electricity. And they realized it is not nearly as fast as electricity. There's a large difference between it, and they wondered why. And they discovered it's because the way that your brain fires is it's both electrical and it's chemical. And then the final one, if a blind person uses one finger to read Braille, the brain area dedicated to that finger expands. Yay or nay? So here, the answer is true. And what that illustrates is something that really only over, I don't even know, they used to think the brain was pretty much set when you're born, and the only thing that happens is deterioration. And we know that that's wrong. There's a tremendous amount of plasticity in your brain. Your brain is changing constantly. Every time you experience something new, you're seeing brain change. And if somebody specializes in a particular area, in that case it's Braille, if you become a piano player, you're a basketball player, you're doing something and you're continually using the same tasks, that area of your brain related to that will get bigger as more connections are made. So there's a tremendous amount of plasticity. Now, one of those questions there, number 6, said we use our brain 10%, or we use 10% of our brain. And we already know, it said that's false. So we know 10% is false. The question is, what's the actual percentage? How much of our brain do we actually use? 10%? Higher, lower than 10? What do you think? So I'm asking.
Maybe simultaneously.
Give me some answers. What do you think? Give me a number. How much? Give me a percent. 15%? Okay, so not that much more?
60.
60%? 5%? Anybody? Yeah.
100%.
100%. Was that where you were going as well?
Yeah.
All right, well, so the answer actually is 100%.
Right.
Now, are we
We'll be
using
brain dead if
100%
you're-
of our brain all the time? No. But there are no areas of your brain that are just like, just a throw on. Where that 10%, and this is a huge fallacy. I couldn't tell you how many times I have heard somebody make the statement, we only use 10% of our brain. And it is just blatantly wrong. Now, why that came up, that's hypothesized, at least from looking back at like the history, it probably came up when early scientists, neuroscientists were studying the brain. They figured that the brain itself does not have pain receptors. So you can remove the skull cap from an individual, and you can stimulate their brain while they're awake, and they won't feel pain. You use a local anesthetic so they don't feel pain from the surgery and the scalp, but they can be awake, and you can stimulate the brain. And they would go in and they'd map it, and they'd stimulate a particular area, and the person would be like, their hand would move. They'd stimulate another area, the person might say, it felt like something touched my skin. They stimulated another area, and the person would report nothing. They stimulate another area, the person report nothing. They stimulate another area, and this guy's like, man, I saw flashes of lights. And when they mapped the whole thing, probably only about 10% of it produced any type of reaction. And so they thought at that time, the other areas just aren't used. That's probably the best hypothesis for where that 10% came. The fact is, though, that even though you don't get an outward reaction, we can now look at your brain in action with scans. And you see it lights up everywhere across your brain. At some point or another, you've got some use. Now, the essential part, how it gets used, is that we are designed with communication in mind. The only way we are able to function is because we have effective communication. There are two major communication systems that play a role in, essentially, in psychology functioning. They are the nervous system and the endocrine system. The nervous system, we're going to spend a lot of time talking about. That's where our focus is. Anybody tell me what the endocrine system refers to? Yes?
It monitors hormones.
Hormones. So the glands that release hormones and the system, hormones travel through your bloodstream and it affects different areas. So that endocrine system is also important. It influences us. But we're not going to focus on that. We're going to focus on the nervous system. This nervous system that we all possess directly impacts how you think, feel, and how you behave. And when we look at the nervous system, and again, I could look at a slice, a piece of brain of a human, and a piece of brain of a monkey. And I could not tell you which is which, because both of them are comprised of the same type of cells. And our nervous system is comprised of two major kinds of cells. Neurons, which are your nerve cells, which serve the major role in communication. And the second type of cells are called glia, or glial cells. Now, as we'll see in a little bit when we talk about the neurons are designed to send signals across the body, to receive signals, to integrate that information. The glial cells, we used to think, again, this is like the advance of science, we used to think that all glial cells were is essentially to provide support. So if you think of what your brain is, your brain's a big old glob of cells. And you've got these neurons that are sending signals. Well, around those neurons, you've got all these other cells that are like holding it in place, giving structure to it. We know they do more than that, though. Not only do they just give structure itself, but some specific glial cells provide insulation for the neurons that increases their communication ability. Some glial cells basically provide nutrients and clean up around the neurons. Actually, within the past probably 10 years, I mean really fairly recently, they discovered that some glial cells actually aid in communication. They talk with the neuron. So we're continuing to gain knowledge. The major communication is happening at the neuron level, at those cells, but those glial cells are extremely important. And you've got far more glial cells than you have neurons. And when you talk about neurons, you've got some estimate trillions of neurons in it. Some argue it's in the upper billions, but we've got a tremendous amount of cells. Now within those neurons, you got all kinds of different types They all share commonalities. And we're going to talk about three major types of neurons. The first type of neuron we'll talk about and refer to as motor neuron. When we're discussing stuff in psychology, motor just refers to as movement. So motor neurons transmit information from the brain, or in some cases from the spinal cord, to your muscles. And your muscles are necessary for you to move. So in that picture, you can see where a motor neuron would be. Motor neuron starts in the brain. Let's say I'm sitting here and I ask you, raise your hand, and you raise your hand. Well, how did you raise your hand? It's because motor neurons sent from your brain, goes down through your spinal cord, into your arm, to the muscles, and allowed you to raise your hand. It allowed that movement to happen. That was motor neurons in charge. We also have sensory neurons. Sensory neurons transmit information from your sense organs to the brain. So as you look up at the screen and you see, the reason you're able to see that information is receptors in your eye detect light, detect distance, detect the different things up there. And neurons from those receptors go to your brain, and basically you get that information and you perceive what happens. Okay? If you think somebody touches you on the back, touching you on the back stimulates sensory receptors in your skin, which sends a message from your back to your spinal cord to your brain, and you register that you got touched. So motor neurons, brain to the muscles, sensory neurons from sensory receptors, ears, eyes, skin, nose, all of that to the brain. And then you have a third general type that are referred to as interneurons, which basically are the interlays between those. So they communicate information internally. If it's not going to your muscles, or it's not going from your sense organs, you're basically dealing with an interneuron. You know, as I said before, these neurons are just cells. Your body's comprised of cells. And hopefully, if you're not in biology now, you remember in high school basic biological functions. And if you think about any type of cell in your body, cells in your body perform certain functions. So at some point you probably learn like respiration, elimination, all those different functions. You've got mitochondria, Golgi bodies or Golgi apparatus. You've got all these different things. Neurons have most of those pieces. Neurons function like other cells, but they're specialized for this internal communication. And so although they do those basic functions, they have more important functions in addition. And again, it's in communication. Now, I will tell you that neurons, there's all kinds of different shapes, sizes, looks, but all neurons share some of the same characteristics. And in this particular diagram here, that yellow cell that you're seeing is a depiction of what a neuron would be. The blue cell that you're seeing, and probably the red one as well, would be glial cells that are around the neuron. And these are so small, and it's hard to really fathom what they're doing. But here's some actual pictures of what specialty neurons might look like, depending on where they're found in the body. Again, we've got all kinds of different shapes and sizes, but no matter where it is, it still has some of the basic structures that allow for this communication. And so we're going to talk about some of these basic structures of the neuron. So here is a typical neuron. And And if you look at this typical neuron, first thing I'll point out, underlined, is we've got what's referred to as the cell body. So within the cell body, the major part that contains the nucleus. The nucleus has your DNA, directs these cells what they're supposed to do. From that cell body, and it is within that cell body that kind of the communication gets integrated a little bit. But branching off of that cell body, you see a long structure that comes out. And when I say long, some of these could be millimeters in length. Some of them can be several feet in length. You have the axon. And it is this axon part that actually is used to send the message. So whenever a neuron sends its signal, it travels down the axon. At the end of the axon, you have what are referred to as axon terminals. Now, if you look at this, again, this simple structure, you could have dozens, hundreds of different terminals. But each one of those terminals is going to terminate in a connection with other neurons. Okay, so message comes down the axon, that message will go all the way to that axon terminal. At the end of that axon terminal, you have a terminal button. And within that terminal button, you have the chemicals that are part of this transfer of information. So there's the terminal button. Those chemicals in the terminal buttons are neurotransmitters. And you all named some of them. Somebody said dopamine, somebody said serotonin. I told you about endorphins. Those chemicals are located in the terminal buttons, and when the message travels down the axon, they're released. And it is that chemical that sends the signal to the next neuron. So within a neuron, it's an electrical process. Between neurons, it's chemical. If you go back and see this particular neuron, the axon is coated with what's called myelin. It's like an insulation. In this particular case, that myelin sheath, each one of those little chunks is a particular glial cell. That type of glial cell is called a Schwann cell. Each one of those is an individual cell. And what that myelin does is just like insulation on a wire. You know how electricity works? It protects, it keeps things in, and it speeds the process. It speeds the message down the line. In between each of those chunks, you have a little gap that still shows the axon. That little gap is called the node of Ranvier. So we're just getting structures down. Coming off the other end of the cell body, you have these branching structures that come off of it. Those branching structures are called dendrites. And it is the dendrites that receive information from other neurons. Axon transmits it, sends a message. Dendrites receive the message. And so although, since we just have a single neuron picture here, where those dendrites are, you'd have terminal buttons coming, terminal branches coming, and connecting all over those dendrites. So each individual neuron would have perhaps thousands of different connections coming in. And those thousands, perhaps, of different connections coming in, if they're sending their signals, this neuron takes that information and within that cell body makes its determination. It says, am I passing this message on? Am I going to fire? Am I going to send this message along? So again, there we've got just the structures themselves. If you can understand that, the simplest way to think about it again is the axon sends the message, the dendrites receive the message, the cell body integrates the information. Now we talk about the actual process. So, I told you it's an electrochemical process. At the end of the terminal button, And this blow up is literally one terminal button. You have the neurotransmitters inside. When the message comes down the axon, electrical signal comes down the axon. It gets to the end and it says, release your neurotransmitter. And where it releases, where one terminal button comes in, end of an axon, dendrites on the other, they do not physically touch. They get really, really close together, but they don't physically touch. There's a tiny gap in between the sending neuron and the receiving neuron, and that gap is called a synapse, a synaptic cleft, a synaptic gap. The synapse, now when that message comes down, the neurotransmitter is released into that synapse, into that gap, and those neurotransmitters cross over and they connect with receptors on the dendrite of that next neuron. Once that connection's made, it activates those receptors, it sends its message, then something happens to those leftover chemicals we'll talk about here later, and then again, the next message, the next neuron decides, is it going to fire? And that's the process we're going to talk about next to see how that process works in terms of firing. So, electrochemical, again, within the structure of the neuron, we've got electrical.
I'm sorry, what's going on?
In between, in that gap, between, we've got chemical. Now, if, in the most basic sense, okay, I'm looking at a single neuron. This neuron is not just connecting to one other neuron. Many other neurons are connected to my one neuron. And my one neuron could connect to many other neurons. But in just this one neuron that we're talking about, single neuron, is this neuron going to send a message? Is it going to fire? When it fires, it's called an action potential. So it's sitting here. And whether or not it's going to fire depends on the messages that are coming in. So if other neurons fire and they send a message to this when they release their chemical, their neurotransmitter, it goes into the synapse, or it goes to the synapse, connects with this neuron. Well, what happens is it changes the electrical charge of my neuron. Okay? If that electrical charge changes enough, if it gets enough message, then it says, boom, I hit it, and now it sends a message down its axon, releases its chemical to the next one, which then they determine, did I get enough stimulation, did I get enough message that I am also going to fire and keep this message going. So again, it's a fairly complex process. That firing, as I said before, is called an action potential.
I know he's saying this is very difficult, but I'm taking like all of the Bible classes and we just talked about this.
And that action potential, a firing of a neuron,
Like,
follows what is referred to as the all or none principle.
You'll be okay. It's next.
What this means is that if a neuron fires, if it gets enough stimulation, enough message that it actually fires, it fires the same every single time. The same intensity, exactly. So like in the diagram here, if you notice the blue is the action potential. And that action potential is identical every single time, no matter how much the stimulation coming in is. So when it fires the same every single time. The question is, does it fire? What's going to determine if it fires? And what's going to determine whether or not it fires is, does it reach enough stimulation to pass what's called a threshold? And if it passes that threshold, it gets enough stimulation, then it's going to fire. And again, every time it fires, it's going to be exactly the same. It's a good analogy for this, to understand this all or nothing principle. is like a pistol. You know how a pistol works, where you actually have to pull the hammer back. Okay? If you imagine, I've got a loaded gun, a loaded pistol. I got the hammer back. It's a cock. Okay? Now, if I think about firing this pistol, if I squeeze the trigger, barely squeeze the trigger, and it is not enough pressure to essentially have the trigger release the hammer, that gun will never fire. I've got to squeeze it hard enough so that once I've squeezed it hard enough, it releases the pin, the hammer comes forward, and the bullet fires. And it does not matter if I squeeze the trigger really hard, the bullet doesn't come out any faster. If I barely squeeze the trigger just enough to make the cock go forward, will the bullet just drop out? No, it's going to shoot. It doesn't matter the intensity of the stimulus. What matters is if it hits enough pressure to release that hammer, it fires. If it doesn't, it's not. And that's the same thing with a neuron. If it gets enough stimulation, it's going to fire. If it doesn't, it won't. Now, how much is enough stimulation? Well, first you have to understand what this electrical part of this is. And where we said a firing is called an action potential when the neuron is not firing, when it is completely at rest. We call it a resting potential. And the resting potential of neurons in your body, the internal electrical charge of that neuron is at approximately minus 70 millivolts. So if you think three-dimensional, think how this works. So imagine, again, two-dimensional picture. It's a little bit harder. Imagine that my arm is the axon, okay? Inside this, there's a membrane that covers the neuron, okay? That membrane is selectively permeable. It allows some things through, doesn't allow other things through, right? Inside, the charge is negative. Inside, the charge is around negative 70 millivolts. What that means is that surrounding that membrane, you have interstitial fluid. You've got fluid surrounding it. You've got other glial cells around it. Whatever, the fluid on the outside of that neuron is positive compared to the charge on the inside. And if you know anything about like magnets, whatever, you know that where opposites attract. Okay? Well, so if you think about this, that neuron, as its just resting state, is negatively charged compared to the outside. Well, there are ions outside. There are positive ions. And in particular, you could probably answer this. If you think about the fluid in your body, or even more specifically, like let's say you go to the doctor, you're dehydrated. What do they give you to rehydrate you?
Pedialyte.
Never been to a doctor or a hospital? If you're like on a drip, what are they actually giving you?
Saline.
Saline. What is saline?
Electrolytes.
Somebody say it clearly. I just hear this. What is saline? Salt water. Salt water. Thank you. Anybody know the composition of salt? NaCl. And when you put salt in water, those ions separate. You have positively charged sodium ions and negatively charged chloride ions. All right? Well, essentially, what's inside the cell, okay, you have a whole bunch of things that are negatively charged. For the most part, what makes it really negative is you have these huge protein ions that are negatively charged that will not pass through that barrier. On the external part of the cell, outside in this fluid, you have sodium ions. And in both places, you've got some sodium ions. In both places, you've got potassium ions. But just in general, the charge on the outside, you've got way more sodium on the outside. Inside is negative. The sodium wants to get in. That positively charged ion wants into the negative. It is drawn like magnets together. but the membrane keeps it out. And so you just got to think constantly when this neuron is not firing. These ions outside are just hammering at the door, trying to get in, but the membrane won't let them. Okay? So we set it up. We've got this resting potential. Neuron hasn't fired. That resting potential, negative 70 inside. Once a message starts to come through, well, what happens is, for my neuron that's at negative 70, it gets stimulated by the neuron. So another message comes in from a neuron. Really what's happening is when that neurotransmitter binds with that receptor, it opens up ionic channels. And either it'll allow some positively charged ions to come in, mainly sodium, or it may do the opposite. It may make it so that positively charged ions escape, or negatively charged ions come in. But something's going to happen to that electrical charge. Either it's going to become less negative, or it's become more negative. And if enough stimulation comes in, and as the stimulation, we'll talk about excitatory messages. So I'm at negative 70, message comes in, opens the channel, and it says, I'm going to allow just a little bit of sodium to come in. Sodium comes in, right at that space. If sodium comes in and it's minus 70, what's going to happen to that internal charge? It'll decrease negativity. Otherwise, it would go from minus 70 to minus 69, let's say. More sodium comes in for more messages. It goes to minus 68, minus 67. If a whole bunch of messages are coming in at the same time, quickly, that charge is going to reach what's referred to as the threshold. And the threshold is approximately negative 55 millivolts. And if that internal charge reaches that threshold, then what happens is there are gates, channels that are closed from that selectively permeable membrane. Once that hits that threshold, those channels open wide. And when those channels open wide, what's going to happen? It fires, but what's specifically ionically what's going to happen? If it was, think of the logic here. If it was a zero charge, inside and outside, and I open up the channel, am I going to have a big effect? If it's equilibrium, you'll just have kind of slow, natural process between the two. But what's actually happening is it's really negative inside. Those sodium ions on the outside are dying to get in. As soon as that channel opens, they flood in. It's like it's negative inside, it is party time. And those ions flood into the cell, which then, where they open, the gates open, it comes in. Well, now, all of a sudden, it becomes positive. And that influences the membrane right next to it, which then says, hey, I hit my threshold, I'm going to open it, which then domino effects. Next one's like positive, open, and it just goes down the line, where you went from resting minus 70, you get enough stimulation, you hit threshold, minus 55, and now all of a sudden, party time, gates open, and you go from negative to all of a sudden now, boom, it's going to shoot up, and it actually reaches a positive charge, to up to positive 40 millivolts in a very short time. Thousandths of a second, what we're talking about. Milliseconds. All right? Visually, this is what it looks like. So if you look at what this diagram is showing you, the x-axis is time, and this is literally in milliseconds, thousandths of a second. So, in number 1, we're at the resting potential. The inside potential charge of this is minus 70 millivolts. As it starts to receive messages from other neurons that are excitatory, then that charge goes from minus 70, minus 69, minus 68, and if it hits the threshold of minus 55, then we have an action potential, a depolarization. Sodium floods in, that goes from minus 55 very quickly to positive 40.
First class is animal
When it
physiology.
reaches that top
We literally just
value
learned
of
about this
positive
today.
40, think now, there's no push for more to come in.
Okay.
The charge is actually positive inside. Now the push would be, I want equilibrium. Positive charge is inside. I want to get out now because it's more negatively charged outside than in. So there naturally goes that way. Your system also has specific mechanisms we're not going to go into, but they have pumps, essentially, that start pumping all those positive ions out. Negative ones are going to naturally come in. And so you see the charge during that 3 repolarization phase go from minus 40 back down to the original. Only it doesn't just go back down to the resting potential. What happens is it overcompensates. Before it catches up, it goes down even below our normal starting resting potential of minus 70. Until then, it comes back up and gets back in line. Now, what's significant about this effect? When that depolarization, that is the action potential. That neuron is fired, sending its message. This only takes thousandths of a second for this to happen. But during that time where it is fired, when we hit number 3, the repolarization phase, in that brief time period, that neuron cannot fire again. That time where it absolutely cannot fire again is referred to as an absolute refractory period. So it doesn't matter how much stimulation comes in at that point. That neuron cannot send another message during that time period. What that means is every single neuron in your body, even if it was flooded with stimulation constantly, it is limited in how many times it can fire. There's always has to be this rest. Have you ever heard the term refractory before? Does that word ring a bell to you? If it doesn't, think back to, I don't know, when did you have health class? 7th grade? 8th grade health? You ever take sex ed in school?
You live in America.
That does not mean anything. So is that a yes or a no? Oh. Yeah, see, I disagree. I guarantee some people in here have sex ed. I mean, it helps. It teaches you. Like you learn about instructions, correct? You understand there's differences, right?
Sex ed, he's talking about sex ed.
Yeah, but what do you say? We have what?
Oh, he's asking if when did we learn about sex ed? 2nd grade, 1st grade, 3rd grade?
Okay? During a male's refractory period, this is where you should have learned this in health education.
I missed it. I missed
For a
everything.
man,
I completely missed it.
once a man has climaxed, he cannot go again. Okay? That's called the refractory period. You should have learned that term. Women, you don't have those. Good for you. Women can have multiple orgasms. Men,
Oh, God.
one, done for a little while.
Oh my God.
Refractory period.
Archer,
Before you
come
go
on,
in, it's the
really?
same thing with a neuron. Exactly the same language. A neuron can't go again during that absolute refractory period. And it doesn't matter how much stimulation comes up.
I can't believe I got that on video.
Now, after that absolute refractory period, when it's coming down during that repolarization phase, it hits that point where it goes past the resting potential and goes even below, more negative than the resting potential. It goes maybe negative 75. Well, that period is referred to as a relative refractory period. Because at that point, it is harder for it to fire again, but it's not impossible. It would just take more stimulation. It still has to reach the threshold of minus 55. So there, it could, if it got the extra stimulation, it could fire then, but it's more difficult, which is why it's called a relative. So if you think about how do we, this is communication. These neurons are firing every time you think of anything, anytime you do something, any action that you engage in. It is because neurons are firing. And if you think if you're hearing really, intricate music or whatever, and you can process all that information, and it starts to speed up the tempo, how can you detect that? No matter how much stimulation is coming in this case, sound waves are coming in, you're limited in how fast those neurons can fire because of that break. How do you differentiate intensity? If every time they fire, it's exactly the same, well, we got this break, what's going to be how many neurons are firing, give you information, how fast they fire, so even if it's constant stimulation, you have a small break, but if you have interspersed music, they'll fire at a much slower rate. So our bodies, our brains are designed to interpret that. How many are firing? Not intensity, they're all the same, but at the rate at which they're firing. And again, they're led by these refractory gears. Now, the way I've described it again, this is just a normal action potential. Every time a neuron fires, it goes through these phases. As it, in an unmyelinated axon, I said it's like a domino effect. So, at the axon hillock, where it begins, comes out of the cell body, that's where these charges are kind of summed together. So it's getting messages from all these different neurons. They're changing the electrical potential. If it reaches minus 55 cumulative, then at the very start of that axon hillock, that channel opens up, sodium floods in. Again, because it comes in, it changes the electrical potential of the area next to it, which then opens up, flooding, opens up, flooding, opens up, and it goes down like a domino. All the way down the axon length, reaches the end, releases its neurotransmitter. Okay? Well, in a myelinated axon, you see a slightly different effect. So in a myelinated axon, you have that myelin, that insulation covering it, and instead of having to go down like a domino all the way the length of the axon, what happens instead is that electrical charge jumps. So at the beginning, axon hillock, as it comes off the cell body, that charge starts, and then it jumps through that insulation and goes through the notoram VA, and it's there. Then the channels open up, and it floods in, and then it jumps, and then it jumps, and it doesn't have to go down that length. So when it's myelinated, it goes tons faster. And if you think of why that matters, anything that you have to do that takes smooth processing, motion, think how coordination you have, okay? The axons that are used for coordination for your movement, they're myelinated. And if that myelin broke down, as it does in multiple sclerosis, that myelin breaks down, individuals' normal movement becomes choppy because it becomes slow, and you have less coordination. And anytime, with age, sometimes you see demyelinization of axons, and it leads to slower reaction times and slower processing speed. So that myelin is extremely important. And again, it just, with that insulation, it just lets it skip along the way. So almost anything, again, where you need speed, you're going to see myelin. And visually, myelin will appear white. So when you talk about like brain tissue, you have gray matter and white matter. That white matter is mainly myelinated axons that you're seeing. All right, so that is the electrical part. Now we're going to talk about the chemical part. So once that electrical potential and action potential comes down, reaches the terminal button, then we have a release of neurotransmitters. And those neurotransmitters work like a lock and a key. So we've got a variety of neurotransmitters. We have not even identified all the neurotransmitters that are in our bodies, but we know a few. And so on the left-hand side of this diagram, we have the sending neuron, terminal button. It's connecting essentially with the dendrite of the receiving neuron. And there's the gap, the synapse between them. So when the message comes down, electrical charge gets all the way in. Basically, it varies, but in this particular diagram, you see there's like, they're called vesicles. There's like sacs that hold the neurotransmitter. What literally happens is when that electrical potential changes, those sacs fuse with the membrane and spill the neurotransmitter into the synapse. Those neurotransmitters then go across and they go into receptors in the dendrite, and they fit like a lock and a key. Each neurotransmitter has a different type of receptor, and unless it is the same, it won't work on that receptor. Once that connects to the receptor, literally what that does is change the electrical potential of the next neuron. It opens some channel. Maybe it's a potassium channel, maybe it's something else. They vary, but it's opening something up and it's changing the electrical charge of the next. And if that one, the next one, receives enough stimulation and changes it to -55, then it's going to fire as well, and it's going to continue the message. And this is happening in, again, milliseconds, thousandths of a second is how fast that happens. Now, in terms of what those neurotransmitters in the synaptic release, synaptic transmission, there are several important steps, pieces that have to take place for this to be effective. And where this really stands out is in terms of when you have problems, or if you think about how do psychoactive drugs work. If you take any type of substance that changes, that affects neurotransmitters, affects the nervous system, it's affecting these neurotransmitters in the synaptic process. And so there's several steps. So the first thing your neuron has to be able to do in order for it to work at all is it has to create the neurotransmitter. That's the synthesis part. So neurotransmitters are chemicals. They have a chemical structure. Oftentimes, it is not just like a single element piece. It's a molecule. It's built up. And so if you think synthesis, you got to have the raw materials somewhere in that neurotransmitter or in that neuron. It's got to put those together. It's got to transport them and get them down to the terminal button so that they're ready to use. So there are some drugs, there are some toxins that might destroy the neurotransmitter itself. If you don't have the basic components, you can't make it. And even if that neuron gets stimulation and fires, when it comes down again, it's like fires blanks, and it won't be able to affect the next neuron if it doesn't have that neurotransmitter there to begin with. Second part is if you have it, and it's there, when the action potential comes down, it's got to be able to release it into the synapse so it can take an effect. And there are some drugs, toxins, environmental things that can block release. That can either stop it from releasing altogether. Sometimes there's chemicals that will destroy the vesicles. So the vesicles usually hold the neurotransmitter. There's holes in them. The neurotransmitter leaks out. When the vesicle fuses, it doesn't release anything. And so that neuron will not have its intended effect because something stopped the chemical process. With binding, I told you the neurotransmitters work with the receptors like a lock and a key. So if you do not have the same neurotransmitter for that receptor, it'll try to go in and it won't work. What also can happen is that receptor site, and you can think of it like visually, you can think of it like each one's a different shape. And each neurotransmitter has a shape that fits into it. That's not really how it works, but metaphorically. Okay? Well, that particular receptor site, there are other chemicals that might also fit into that receptor site. Some of those other chemicals that fit in there might do just like the original neurotransmitter. It might turn the lock and work and cause it to do what it is supposed to do. Other ones might fit in there, but it's like a blank, it's like a slug. It doesn't actually work. It blocks the receptor site, but it doesn't produce the intended effect. So that when the real neurotransmitter is released, it doesn't have a receptor site to go to, it's blocked, and it keeps it from functioning. Inactivation. So in this process, I said neurotransmitter is released, goes into the synapse, goes into the receptor site, produces an effect, and then it's released from the receptor site. It's sitting in the synapse. One of two things will happen. If neither of these things happen, think what would occur. This neuron's fired, it releases its neurotransmitter, it's did its job, and now it's sitting there. What it would naturally do is go into the receptor site again, get released. Go into the receptor site again, get released. And it would just keep doing what it's intended to do infinitely, I suppose, if nothing happened. But instead, what happens is one or two mechanisms. One is inactivation. You have enzymes within that receptor site or within that synapse. And some of those enzymes are designed to destroy the excess neurotransmitter. It's released, it does its job, now it's sitting there, and it gets catabolized, it gets broken down into its components, which then maybe can be reused to make others. That would be enzymatic degradation or inactivation. The second thing that can happen is, after it does its binding, it's released, it's sitting there in the synapse, the neuron that released it can suck it back in to use again. That's called reuptake. So frequently you will see release, the neuron that sends it, sucks it back in to reuse again. Now, probably some of the drugs, psychoactive drugs, that you are most familiar with, work through that process. So like if you just named a random drug that treats some psychological disorder, what comes to mind? Give me a name of one. I actually don't know that one. Yeah. Zoloft. Okay, Zoloft is a good example. So Zoloft is a drug that fits into a category of drugs that are called SSRIs. Selective serotonin reuptake inhibitors. From that name, can you tell me how it works? Say again. It blocks reuptake. So if you think about it, that neurotransmitter is released, it binds, it's sitting there in the synapse. Normally, it gets sucked back up, but this drug keeps it from getting sucked back up, which means it stays in the synapse, and it'll keep binding. It leads to an effective increase in the neurotransmitter in the synapse, which then, if you think logically, if that drug leads to an increase in serotonin in the synapse, and it's an antidepressant, what might be, and I put might largely, logically, what might be the reason individuals are depressed? Is it because they have too little or too much serotonin? Use logic here. What's the action of the drug going to do? It blocks reuptake. Does that lead to an increase or decrease in the neurotransmitter in the synapse? An increase. If it blocks reuptake, it makes more there. If it alleviates depression because there's more available, logically you could go, well, maybe it's because you don't have enough serotonin for individuals who are depressed. This drug increases the amount of serotonin. Now, we know that it's not that simple, but there's one hypothesis that says that's what depression is related to, is an inability to effectively use serotonin, and if we can increase the amount of serotonin, it should alleviate the symptoms. But again, it's all in this part. This is the basic level of how this neuron works. And this is every neuron that's in your body. So, we've got the basic structures. Now we're going to talk about some of the specific neurotransmitters. So I said, we know some. We definitely don't know all. One of the most highly studied neurotransmitters that we do have a pretty good idea about is referred to as acetylcholine. Now, acetylcholine in the brain, we know is associated with memory processes. It's associated with learning. But where I first learned about acetylcholine, and where we know and can easily study the effects of acetylcholine, is acetylcholine acetylcholine is the neurotransmitter that allows you any type of movement. So when we talk about motor neurons go from the brain, they come down, they meet muscles to tell the muscles to move. Well, the neurotransmitter that is released in motor neurons is acetylcholine. And how I say, when I first learned this, when I was an undergraduate, many years ago, I taught anatomy and physiology lab. And I remember in one of our labs, when we were in anatomy and physiology, we isolated a frog's leg, and you had the nerve from the frog's leg, and we had a little stand, and you had the frog's leg hanging on the stand, you had the nerve, and we put it on a slide, and if you poured acetylcholine onto that nerve, anybody want to tell me what happens? The leg starts kicking, okay? You put that neurotransmitter directly on that, it is going to start kicking. If you then took a different chemical, and I'm blanking on the name of the chemical now, you should know it, that we would put on there that would block the receptor site for acetylcholine, it would immediately stop. So acetylcholine, we know, for any type of muscular movement, that is the neurotransmitter. And I use that, again, it's easy. We can isolate nerves in your outer body. It's harder to do stuff in your brain. We can isolate animals, use the same neurotransmitter there, and we can do experiments to show how this works. And it's also a good one because there's several other chemicals that affect the acetylcholine system in different ways. Some of those work the same way that acetylcholine would. And if you have another agent, another chemical, that basically produces the same effect, it works as if it was that neurotransmitter, it works in the same way, we call that an agonist. An agonist works in the same way. Now, I say in most cases it excites it because it makes it do the same thing, but there are some neurotransmitters that are inhibitory. So, that's why it says it works in a similar way. You have others that produce an opposite effect or block the normal use. Those are called antagonists. So they inhibit the normal functioning of that neurotransmitter. And so we're going to talk about 4 different chemicals that are both agonistic and antagonistic towards acetylcholine, some of which you probably heard of. And we'll talk about these effects. So the first one, I guarantee you all heard of, and that's nicotine. You know, nicotine is the active component, the active drug in cigarettes. And nicotine, the chemical structure of nicotine matches the chemical structure, the shape per se, of acetylcholine. So this sits in the same receptor sites. So even though it's a different thing, it's a different chemical from acetylcholine, it fits into that same receptor site. And so if you smoke cigarettes, you chew tobacco, what are the effects that are happening, the psychoactive effects? Well, one is nicotine actually increases arousal. It increases attention. Because it's getting in receptor sites in the brain that are associated with attention, and it perks you up. Okay? So it is an agonist for acetylcholine, and its working mechanism is because it fits into the same sites. So it stimulates those same receptor sites, gives you a similar effect. Now, a drug that you may not be familiar with, unless you studied anthropology, is curare. And curare, I remember learning about curare when I was an undergrad taking anthropology, cultural anthropology. Curare is a poison, it's a toxin. South American tribes will use it to cover their arrows or their spear points when they're hunting. It is an ACH, acetylcholine antagonist. And so when they fire their bow and it hits an animal, it gets in the animal's system, gets in their bloodstream, gets in eventually to the receptor sites, and as an antagonist, you want to tell me me what curare is going to cause? Paralysis. So it does not allow ACH to work. It actually blocks those sites. So it fits in like nicotine did. It fits into the same site, but instead of turning the key and making it work, it blocks it so that when ACH is actually released, it can't get in. And so it causes the animals paralysis. Eventually the paralysis actually gets to their diaphragm, so they suffocate. But boom, they're not moving anymore. They've got their meal. Now, again, you probably also heard of Botox or botulism. Botulism is a toxin that also causes paralysis. And if you have botulism poisoning, you don't get treated quickly, you're going to die. And yet, we have a whole industry of individuals purposely injecting botulism toxin into their faces. Why would you do that? What's it doing when you inject Botox into your skin? It is an antagonist, the ACH. So what is it doing? It is paralyzing your facial muscles. That's exactly what it's doing.
So that's what.
And so it makes your wrinkles go away because those muscles are paralyzed, basically. And if you do not know, I mean, I would never recommend anybody doing Botox. For one, be who you are. For two, you can die from it. It is poison. And then, what you should never try, the last one we'll talk about is black widow spider. Black widow spider, the venom that it produces gets into your body. It also affects ACH. It acts in a different mechanism, though. Instead of working at the receptor site, what black widow spider does is it causes an increase in release of the neurotransmitter. So you get bit, and all of a sudden you get a surge of ACH into your system. Anybody tell me what those symptoms would then be? Not seizures per se. Seizures we usually think of directly from the brain, but absolutely you're going to have muscle spasms because you got this unfettered release of ACH. And so that is one of the symptoms of those. So again, we've got 4 different chemicals. This is just an example. All these chemicals work on that same system. They affect it in different ways. Some block the neurotransmitter, some increase release, some activate as the same way. So we can name any neurotransmitter system that you talk about, and it can be affected by any of those different steps that we talked about in terms of synaptic transmission. Now, some of the other neurotransmitters that we have studied, that we do not know everything by any means, and I don't want you to give that impression of what these neurotransmitters do. What researchers have been able to discover is that a certain type of neurotransmitter, we can find areas in the brain where that neurotransmitter is prolific, where it's used mostly. We can see what that area of the brain is associated with, and so we can say this neurotransmitter must be associated with these things. So one of the other neurotransmitters, somebody named earlier, is dopamine. We know dopamine is associated with several functions. One is also movement, only in a different pathway. So instead of where ACH is related to movement directly at the muscular level, for dopamine, you have a tract in the brain that is mainly a dopaminergic, uses dopamine tract, that is also associated with more coordination and movement. And so if you have Parkinson's disease, everybody knows what Parkinson's disease refers to. Parkinson's disease has to do with you don't have enough dopamine available. And so you have poor movements. In this case, what they treat, well, no, I ask you all, if anybody has studied this. Anybody know what the treatment is for Parkinson's disease? Nobody? So the most usual treatment for Parkinson's disease is a drug called L-Dopa. L-Dopa is a precursor to dopamine. And the reason individuals have Parkinson's disease is they can't produce enough dopamine. It's not something that crosses the blood-brain barrier, so they have to give them this drug that gets them the basic line, and then they'll create their own, which then helps their movement. We know dopamine is also associated with learning, with attention. It's associated with those pleasure centers in the body we talked about, or in the brain. So when you think about addictive drugs, dopamine usually comes in. Dopamine has also been implicated in schizophrenia. So there is a dopamine theory of schizophrenia we'll talk about later in the semester. Norepinephrine is another neurotransmitter. Norepinephrine is also called noradrenaline. And adrenaline and noradrenaline are both neurotransmitters and hormones. When their hormones are released into the bloodstream, go to the glands and deal with the endocrine system. When they're in the brain, they're neurotransmitters. And norepinephrine is associated with alertness and arousal. We know the centers that are associated with wakefulness and stuff. Those are mainly norepinephrine. Somebody earlier mentioned serotonin. We know serotonin is related to mood, for one. It's also implicated with hunger, with sleep behaviors. And as I try to get you to logically think through, depression and anxiety are also seen as somewhere in that system, serotonin plays a role. Fairly unique neurotransmitter is GABA. And why I say this is fairly unique is that of all the neurotransmitters, I'll tell you, depending on where in the brain or in the body they are, most neurotransmitters can do either increase the chance that the next one's firing, they excite, or they can inhibit, they can decrease the chance. Most of them are excitatory. GABA is solely inhibitory. So when GABA is released into the synapse and it binds with receptors, instead of increasing the chances that the next neuron's going to fire, it decreases the chances that the next neuron's going to fire. And if you think logically again, where would this come into play? If you're trying to slow the system down instead of speed it up, if you're dealing with anxiety or seizures, where you're trying to slow the system down, GABA has been implicated in that. There's not enough GABA to slow the system down, and so you have too much activity. Glutamate, as far as we know, is the most prolific neurotransmitter. There's more glutamate than any other neurotransmitter. We know memory is associated with glutamate. But again, when we talk about memory is very complex, so we know that one is one of the neurotransmitters that's important. And then the last one that we'll discuss are endorphins. And again, endorphins are your body's natural pain reliever. So if you've ever heard of like a runner's high, or if you've ever been in an accident, you damaged something. So like for instance, I can remember when I was in grad school, I was playing softball, and I tore my ACL. Running home, knee slammed in, I flipped over it immediately. I'm just like, it's gone. I'm screaming, in a tremendous amount of pain. They take me off the field. 5 minutes later, I'm like, man, I'm ready to go. I mean, I couldn't even stand. I'm like, I'm ready to go. But I was like, I felt awesome because it was like getting flooded with an opioid